Imagine a microscopic library, a vast collection of knowledge stored within the tiniest of spaces. This library holds the instructions for building and maintaining a living organism, a detailed guide to life itself. This is the essence of DNA, the blueprint of life as we know it, a molecular code that defines every living being. DNA's structure resembles a twisted ladder, the famous double helix, a marvel of biological engineering. The rails of this ladder are made of sugar and phosphate molecules forming a sturdy backbone. Connecting the two rails are rungs formed by pairs of nitrogenous bases, adenine, thymine, guanine, and cytosine, the building blocks of genetic code. These bases always pair up in a specific way, A with T and G with C, ensuring the accuracy of genetic information. This base pairing is crucial for DNA replication, allowing cells to divide and reproduce accurately. Think of DNA as an instruction manual, a comprehensive guide to the development and functioning of an organism. Each sequence of bases represents a specific instruction, a unique directive for cellular processes. These instructions determine traits like eye color, height, and even susceptibility to certain diseases, shaping the diversity of life. The complete set of DNA instructions in an organism is called its genome, a comprehensive map of genetic information. The human genome contains roughly 3 billion base pairs, highlighting the immense amount of information encoded within our cells, a testament to the complexity of life. It's a complex and elegant system for storing and transmitting genetic information, a sophisticated network that ensures the continuity of life across generations. Cells are constantly dividing to create new cells. This process is essential for growth, repair, and reproduction. Before a cell can divide, it must first duplicate its DNA. This ensures that each new cell receives a complete set of genetic instructions. This process of duplication is called DNA replication. It is a fundamental process in all living organisms. DNA replication is incredibly precise. Errors during replication can lead to mutations. These mutations can be harmful, beneficial, or have no effect at all. Over time, accumulated mutations contribute to evolution. They allow populations to adapt to changing environments. DNA replication begins with the unwinding of the double helix. An enzyme called helicase is responsible for this task. Imagine helicase as a zipper, unzipping the DNA molecule by breaking the bonds between the base pairs. This creates a replication fork, a Y-shaped region where new DNA strands are synthesized. The separated strands serve as templates for creating new complementary strands. The unwinding process is crucial. It exposes the nitrogenous bases that serve as templates for the new DNA strands. This ensures that each new DNA molecule is an exact copy of the original. Once the DNA strands are separated, the replication process can begin in earnest. Another enzyme called primase comes into play, initiating the next crucial step in DNA replication. Primase is responsible for laying down a short sequence of RNA nucleotides called a primer. This primer is essential for the next phase of replication. This primer acts as a starting point for DNA synthesis, providing a necessary foundation for the process. DNA polymerase, the enzyme responsible for building the new DNA strand, can only add nucleotides to an existing strand. Without the primer, DNA polymerase would have no starting point. The primer provides a 3' end for DNA polymerase to attach to, which is a critical aspect of the replication process. This is crucial because DNA polymerase can only add nucleotides in the 5' to 3' direction. The directionality ensures that the new strand is synthesized correctly. The primer is like the foundation of a house. Just as a house needs a strong foundation to be built upon, the DNA strand needs a primer to begin synthesis. It provides a base upon which DNA polymerase can build the new DNA strand, ensuring that the genetic information is accurately copied and passed on. Understanding the role of the primer and DNA polymerase is fundamental to grasping how genetic information is faithfully replicated in every cell division. Section 5. Building blocks of life. With the primer in place, DNA polymerase begins its work. It reads the sequence of bases on the template strand and adds complementary nucleotides to the new strand. Remember, A always pairs with T, and G always pairs with C. This ensures that the new DNA strand is an exact copy of the original. DNA polymerase moves along the template strand, adding nucleotides one by one. The energy for this process comes from the nucleotides themselves. Each nucleotide carries 3 phosphate groups. The breaking of bonds between these phosphate groups releases the energy needed for DNA polymerase to function. Section 6. Leading versus lagging strands. In this section, we will explore the fascinating differences between the leading and lagging strands during DNA replication. DNA replication is not a symmetrical process. It involves a complex interplay of enzymes and structural components to ensure accurate duplication of genetic material. The 2 strands of the DNA molecule are antiparallel, meaning they run in opposite directions. This antiparallel nature is crucial for the replication process. This poses a challenge for DNA polymerase, which can only synthesize DNA in the 5' to 3' direction. This directional constraint necessitates different strategies for each strand. On one strand, called the leading strand, DNA polymerase can move continuously, adding nucleotides one after another. This continuous synthesis is efficient and straightforward. On the other strand, called the lagging strand, DNA polymerase must work in fragments. This discontinuous method is more complex and requires additional steps. These fragments, called Okazaki fragments, are synthesized discontinuously. Each fragment is initiated by a short RNA primer. They are later joined together by another enzyme called DNA ligase, which ensures the integrity and continuity of the lagging strand. This difference in replication between the leading and lagging strands arises from the directional limitations of DNA polymerase. Understanding these mechanisms is essential for comprehending how genetic information is accurately passed on during cell division. Section 7. The role of DNA ligase. After DNA polymerase has synthesized the new DNA strands, another enzyme called DNA ligase comes into play. Ligase acts like molecular glue, sealing the gaps between Okazaki fragments on the lagging strand. This creates a continuous strand of DNA. The action of DNA ligase is crucial for the integrity of the newly synthesized DNA molecule. It ensures that both the leading and lagging strands are continuous and complete. Section 8. Proofreading and repair. DNA replication is a remarkably accurate process, but errors can still occur. To minimize these errors, DNA polymerase has a built-in proofreading mechanism. It can detect mismatched base pairs and correct them. In addition to DNA polymerase's proofreading ability, cells have a variety of repair mechanisms to fix damaged DNA. These mechanisms help to maintain the integrity of the genetic information. They ensure that errors during replication or damage from environmental factors are quickly repaired. Section 9. The importance of replication. DNA replication is essential for life as we know it. It is the foundation of heredity, ensuring that genetic information is passed down from one generation to the next. Without DNA replication, cells could not divide, and organisms could not grow or reproduce. DNA replication is a complex and fascinating process. It is a testament to the elegance and efficiency of biological systems. Understanding DNA replication is crucial for advancements in medicine, agriculture, and biotechnology.